Very good evening, students. Very good evening. Oops, let me change my. Is my voice is okay today? Earlier you were saying the voice was not fine. Uh, Please confirm me that voice is okay and screen is being shared clearly and all is good. Only then we will continue further. Some of you students, one or two, confirm me that all is fine, sound is okay, and screen is being shared fine, and we are good to go. So I have not much time to say good words as a welcome and everything. I have to straightforwardly start because already 10 minutes are gone and we have to do a lot today and quite exciting things actually. Let me see. There's some problem with the recently, okay, virtual labs. Okay, they will come in spring 2021. So this will not work. Fine. No worries. What else we have? Do we have this one? So we used to have these basics techniques in virtual form, but recently Adobe Photoshop has some changes, and we cannot have these labs, unfortunately. Yes, we cannot have these labs. Anyhow, it's not working. Anyhow, so not wasting much time, we start with our molecular method state. So I also shared on the Telegram some two fundamental techniques in cell culture. If somebody wants to go in details about cell culture, could read these performers. They are by Sigma Aldrich, a very known company in the field of cell culture. So all things how the morphology should look like: HeLa cells, BA cells, endothelial cells, fibroblast, neuronal cells, what kind of culture do we require, what kind of morphology. So it's quite in detail. Just go through them. So it will be quite good one. Um, so where I was, yeah. So today we will deal with the basics of your molecular biology techniques today. So we try to cover as much as possible. If some are left, we will try to do them tomorrow. Basically, in this part, today topic will be divided into, first we will deal with the PCR, that is your polymerase chain reaction, then gel electrophoresis, then your blotting techniques. Then further we go for gene expression analysis like real-time PCR microarrays, then some recombinant DNA technology techniques like cloning of DNA fragments, Sanger sequencing and next generation sequencing. Then method to study gene function, cell culture methods, generation of transgenic mice and knockout mice. So basically in this part we are dealing with most of the techniques that are majorly used whenever freshly you're going into a lab. So if you are aware of these techniques before gonna start your master thesis or PhD, you will be one step ahead. So it will be nice if you work on the assignment part also on this, because you go all through them and try to pen down with your paper or use with the help of WordPad thing. So first thing first, PCR. Before I start, I want to just check all is well. Live streaming is good. Okay. बच्चे Rishanku Pinea, write me in personal message. I will let you know because if I start speaking about that, our whole lecture will go away. So I will be now specific to the topic. So let's start. So PCR, that is your polymerase chain reaction. Basically, what we do, we want some specific DNA, some specific part of DNA. So we have this, long your helical structure of a DNA, and I'm looking for a specific part in between. Let's say from here till here, yeah, and it has some particular sequences like C, T, T, uh, G, C. Let's say we have uh, five amino acids, and then I want to have only these five base pairs of your nucleotides to be available in huge amount. In lot of amount, I require them for any forensic reason or for any my reactions, gel electrophoresis. There could be many applications of having a huge amount of DNA, and so for that, PCR plays a role. And PCR is nothing, it's just a thermocycler actually. In this, we raise the temperature, then we decrease, and then a little bit increase. And on basis of that, you will have lots of DNA primers, and at the end, you have billions of primers after the reaction is over. So, first thing first, so can be used to amplify rare specific DNA sequences from complex mixture when ends of the sequence are known. So, when we know the end of the sequences, we can create the same base pairs in huge amount. So, PCR amplifications of mutant alleles allows detection of human genetic disease. So, we can detect many human genetic diseases like Alzheimer or anything like that. And DNA sequences can be amplified by PCR using in cloning as a probes or in forensic science also. So Cary Mullis, remember this name, Cary Mullis. He got the Nobel Prize winner for chemistry in 1993. It's not long. So I was born in 1987. So after my 5 years of birth, this PCR was invented, and Cary Mullis got the Nobel Prize winner for his invention. So basically in PCR as a recipe, so in any proteomics or in any other techniques, you have, you know, some recipe. And for a recipe, you need some ingredients. If you want to make biryani or shahi paneer or any dish, so we all know that to prepare that we need some ingredients specifically in specific amount, and we have to add them in specific orders one by one. And for the same, sorry for that sound, you have buffer, you have template DNA, primers, nucleotides, DNA polymerase, and water. So these are the six things that you require for your particular PCR reaction. And then it initiated. So what will happen? You have a target sequence here. In the initial part, we will have the denaturations. We heat it with around 90 degrees Celsius to separate the DNA strands. Then we bring to the 50 degrees Celsius, we cool it, and here our primers will attach and start producing corresponding DNA and making a hydrogen bonds with it. Then the same will be extended with the help of DNA polymerase with the 3' end of each primer. So you have new nucleotides at extension. So from 50 we increase the temperature to 72 degrees Celsius. So here's around 50, and it's more than 50 here. It's more like 90 or something. I'm not sure, maybe it's 90. Then it goes further into cycle 2. So same reaction of your step 1, 2 and 3, they will be repeated in the cycle 2 with 4 molecules. Then the cycle 3 will yield 8 molecules with 2 molecules in these white boxes. These are the target sequences that you are looking for. So like this, many cycle will be goes on, and hence at the end we have our PCR product. So that was so maybe I could find some uh, 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 To make you understand more about PCR, I have to show you one virtual lab of it for sure.
CSI Lab, which explores the principles of polymerase chain reaction and gel electrophoresis by taking students through an investigation of a murder mystery. Investigating the crime scene, the student collects blood samples and hopes that the murderer has left traces of DNA. After sampling, the student accesses a virtual lab to perform DNA analysis. In the lab, a PCR kit, purified DNA from the crime scene, and a full lab bench setup are available.
Okay, sorry.
It is now up to the student to mix the correct reagents and perform PCR on the PCR machine in the lab.
This is not for you. So actually, we used to have this link working to perform virtual lab, but unfortunately, it will come in spring 2021. But I will still share the link with you in the future if it is coming, so that we can learn about it. But for those, some of you might be aware of how PCR is working, but for those not, I want to play one virtual video for the same. I think that one was not good one because it was selling us. Maybe this one will help us.
In this experiment, we'll carry out several steps to complete the conventional polymerase chain reaction, PCR, using 3D Biology Virtual Lab from CSI Labs. Log in to your CSI Labs account using your username and password, and then go to the lab to conduct the experiment. If you're watching this video on our YouTube channel, please make sure you subscribe first and activate the notification bell to get our new videos regularly. The goal of the PCR experiment is to amplify a targeted part of the DNA to a size that may reach up to 10,000 nucleotides, which will allow getting millions of copies of the targeted DNA strand in a short time. For this, PCR has gained popularity for a wide range of applications, such as the detection and analysis of some dangerous viruses such as COVID-19. Also, molecular biologists use PCR in gene cloning and DNA sequencing, and forensic scientists use PCR to connect blood, saliva, or tissue left at the scene of a crime. The Virtual Biology Lab from CSI Labs provides you with an interesting interactive experience where you'll be able to conduct the experiment through 3D simulation that includes all the devices, tools, and materials used in the biological laboratories. Let's start the experiment. The PCR experiment is divided into 3 main steps. First step, prepare an amount of the master mix sufficient to 6 samples of the DNA by adding the special reagents for the experiment in the master mix tube. 243 microliters of distilled water. 30 microliters of the 10x PCR buffer containing 6 microliters of the 10 millimeters DNTPs. 6 microliters of the forward primer. 6 microliters of the reverse primer. 3 microliters of the Taq DNA polymerase. Second step, liters of the master mix you just prepared to 1 microliter of each DNA sample. Third step, place the Eppendorfs in the thermal cycler. Then adjust the settings as following. Initialization denaturation stage at 94 degrees Celsius for 5 minutes for 1 cycle. Multiplication cycles stage, which is divided into denaturization step at 94 degrees Celsius for 30 seconds, 30 cycles. Annealing step at 50 to 65 degrees Celsius for 30 seconds, 30 cycles. Elongation step at 2 degrees Celsius for 1 minute, 30 cycles. Final elongation at 72 degrees Celsius for 10 minutes